now we will move on to highlighting a cross-platform collaboration that was a proteogenomic analysis featuring Adam Resnick from the Children's Hospital of Philadelphia, Dr. Francesca Petralia from the Icahn School of Medicine at Mount Sinai, and Dr. Pei Wang from Mount Sinai as well. So with that, I will make Francesca the presenter.
Thank you, Valerie. Do you see my screen? Yes. Yep. Can you hear me? Okay. So it's a great honor to be here today and present this work that was a great collaboration between Kids First, the CPTech Consortium, and the CBTN Consortium. So this is the result of a great collaboration, and it's an honor for me to represent the work of so many scientists. So the title of the presentation is Insights from Proteogenomic Characterization of Pediatric Brain Tumor, and this paper was recently published in Cell. So I will give a quick background. So pediatric brain tumor are the leading cause of disease-related death in children and represent distinct biological entities from adult brain tumor defined by the developmental context. So there is a need for precision medicine approaches limiting the toxicities for the developing brain. So pediatric tumors are very different from the adult tumor because they have much lower mutation burden. It's for this reason that it's key to leverage proteomic data in order to direct therapeutic strategies. It's for this reason that CPTech and CBTN and Kids First came together in order to realize to generate the first proteogenomic data of 7 different histologies of pediatric brain tumor. And again, the paper was recently published in Cell. So I think everyone here is familiar with Kids First and CBTN, so I will just spend a few words about the CPTech. So CPTech is a consortium of the NCI and is generating large-scale proteogenomics data for different cancer types for the first time in order to accelerate our understanding of this deadly disease. So this is the first time that CPTech collaborated with CBTN and Kids First to generate the first proteogenomic data of 7 different histologies of pediatric brain tumors. So, so as you can see from these slides, there are so many institutions that were involved in this paper, and the collaboration was key in order to create such a work. So the goal of this paper was to generate proteogenomic data and provide a comprehensive molecular characterization of 7 different pediatric brain tumors, then link the genomic aberration to proteomic profile, and delineate novel insights that were uniquely provided by the proteomic data. So this is the overview of the data. So we have more than 200 pediatric brain tumor samples. We have for 18 pair of sample, we have initial and progressed data. And we did whole genome sequencing data. So we have mutation and copy number variation data, RNA sequencing data, and then we did global proteomics and phosphoproteomic profiling of thousands of proteins. So as you can see, so we have 200 pediatric brain tumors spanning 7 different histologies. So for some histologies like ATRT, craniopharyngioma, we have a lower number of samples. But of course, I mean, this is the first time this data was generated, so it's really informative and really can provide great insights. So first, what we did is to cluster samples from different histologies based on proteomic profiling. And we identified 8 different clusters, and some of them were a mixture of different diagnoses, as you can see from those pie plots. I hope you can see my my pointer. So one of the cluster was named aggressive because basically contained ATRT, medulloblastoma, high-grade glioma, so more aggressive subtypes, and a subset of ependymoma. As you can also see from the Kaplan-Meier curve, these aggressive subtypes was resulting in a worse prognosis. Another cluster that was really interesting because it was a mixture of different subtypes of different diagnoses was the cranio LGG BRAF V600E cluster that was a mixture of low-grade glioma carrying the BRAF V600E mutation and a subset of craniopharyngioma. So this cluster was actually very interesting, and we were surprised that those craniopharyngioma that were not carrying the BRAF V600E mutation were clustering together with a subset of low-grade glioma carrying that particular mutation. So I want basically to focus on this splitting of craniopharyngioma into these two subtypes. So as I said, so the craniopharyngioma were split in one cluster that was enriched of low-grade glioma carrying the BRAF V600E mutation, and another cluster enriched of enriched of low-grade glioma BRAF wild type. So these subtypes of the craniopharyngioma was only captured by proteomic and phosphoproteomic data, because if we look at the clustering based on RNA data, the craniopharyngioma were split in one, were basically allocated to the same cluster. Instead, the proteomic and the phosphoproteomic, they basically separated consistently the craniopharyngioma into these two subtypes. So why this is important? Because craniopharyngioma currently has no effective chemotherapeutic option, and BRAF V600E mutation has been observed in adult, but not in pediatric craniopharyngioma. So this proteomic-based clustering actually revealed that a subset of craniopharyngioma not carrying a BRAF V600E mutation has some similarity based on proteomic and phosphoproteomic profiling to the low-grade glioma carrying that particular mutation. We know that MEK inhibitor has been shown to be effective for low-grade glioma BRAF V600E patient and is being tested for adult BRAF V600E craniopharyngioma. So MEK inhibitor treatment could be beneficial for this subset of craniopharyngioma, so pediatric craniopharyngioma, not carrying BRAF V600E mutation, but showing a consistent proteomic profiling to the low-grade glioma carrying that particular mutation. So we did two validation in order to make sure that this signal was real. So the first one is based on the kinase activity, based on the phosphorylation data. And the second, we ran an independent MRM panel which confirmed the splitting of this craniopharyngioma into two subtypes. So for the kinase activity, what we did is for each kinase, we know which phosphosites are targeted by each kinase. And based on the phosphorylation abundance of those phosphosites targeted by a particular kinase, we construct a signature. And basically, as you can see from this heat map, we showed this kinase activity score for different kinases, AKT1, mTOR, MAPK1, for the craniopharyngioma samples, where at the top you have annotated the craniopharyngioma in yellow, the craniopharyngioma allocated to the cluster enriched of low-grade glioma carrying the BRAF V600E mutation, while in purple, the craniopharyngioma allocated to the other cluster enriched of low-grade glioma BRAF wild type. And as you can see, AKT1, mTOR, MAPK1, and MAPK3 were upregulated in the first subset, as also shown on the diagram on the right side. So basically you can see that those kinases that are downstream of MEK, they're upregulated in the set of craniopharyngioma clustering with the low-grade glioma carrying the BRAF V600E mutation. And then we also ran this independent MRM assay to validate the signal. And what we show here is that, for example, if we focus on MAPK1, that is, of course, a kinase downstream of MEK, ERK3, you can see here the box plot showing the our TMT data and also the mrmsa, and you can see that there is a clear separation between the two groups. The craniopharyngioma clustering with the LGG BRAF V600E mutation in yellow, and the LGG and the craniopharyngioma clustering with the BRAF wild type. So now, as a next step, there is an effort to test the MEK inhibitor therapy in a clinical trial that was started in collaboration with the Pacific Pediatric Neuro-Oncology Consortium. Another thing we did in the paper was the immune landscape. So based on the bulk RNA data, we estimated the fraction of microglia, macrophages, different immune cell, neurons, astrocytes, and we clustered sample based on this immune profile. So what we found are 5 different clusters. One is the hot tumor that has the higher infiltration of microglia and macrophages, and you can see that actually contain different histologies, so low-grade glioma, high-grade glioma, ganglioglioma. And then we have the epithelial cluster that is basically containing all the the craniopharyngioma sample. So we capture here the cell of origin for sure. But what I want to emphasize here is that we found PD-1 and CTLA-4 upregulated in this particular cluster. So an anti-PD-1 therapy might work for the craniopharyngioma samples. Then we have the neuronal group that has a higher infiltration of neurons, or genes in neurons, highly expressed in this group, and lower immune infiltration than the hot tumor. And then we have the so-called tumor that has low immune infiltration and higher cell cycle and higher proliferation. And basically, as you can see, it's basically medulloblastoma, ATRT, and ependymoma located to this cluster. So here it's just a diagram showing what we found for each immune subtype. And what we did is an extensive pathway analysis based on RNA, protein, and phosphoprotein data. And what we found is that some of the pathways were only upregulated in a particular subtype based on the proteomic data and not captured at all in the RNA domain. For example, if we look at the cold tumor, we found signaling by Wnt and beta-catenin upregulated in cold tumor. These are well known to be immune inhibitor pathways and were captured only in the proteomic domain. Or in the neuronal group, we found upregulation of OXPHOS, glycolysis, mitochondrial that was only captured in the proteomic and phosphoproteomic domain. And again, so for the hot and the epithelial, the pathways were more consistent across different data types. And then we also looked into the effect of mutation on protein expression. Of course, for pediatric, we have less mutation burden, but of course, there are some key mutations, for example, BRAF, as I said, in low-grade glioma. And as you can see from this plot, the BRAF mutation was affecting the proteomic, the global proteomic data more than the RNA level data. And the same for beta-catenin in craniopharyngioma and NF1 in high-grade glioma. We also did a CNV cis regulation. It's just for the sake of time. Of course, I have decided to include these things, but of course, we linked all the genomic data we had with the protein and phosphoprotein data. Then we looked also at the trans effect of beta-catenin in beta-catenin mutation in craniopharyngioma, and again we found some pathways that were altered in the proteomics domain, but not in the RNA. For example, Wnt signaling or coagulation. So we were really interested to understand the difference between protein and RNA data and what really the proteomics data could contribute to the overall picture. Another interesting point we did is the survival analysis for high-grade glioma. What we found is a two marker, IDH1 and the 2 protein expression were associated with adverse survival outcome in H3 wild type high-grade glioma. So we know that IDH1 and 2 mutation is relatively rare in pediatric high-grade glioma, and also for our data. But we found this prognostic value of IDH1 and 2 protein abundance for pediatric H3 wild type. So in order to confirm this signal, because the sample size was a little bit limited, we had only around 30 sample for high-grade glioma, we validated this signal with an additional 23 high-grade glioma H3 wild-type tumor. So these are the results for the discovery cohort and the validation cohort. So what we found is that a higher IDH1 and 2 protein abundance correlate with better survival in H3 wild type high-grade glioma. So you can see from this plot the hazard ratio of the combined score capturing IDH1 and 2 global abundance in H3 wild type has a significant association with survival, and the same for this validation data set. As also is shown here by the Kaplan-Meier, so you can see that H3 mutant is the one with the worst prognosis, as already known, and then we have the H3 wild type with high ADH1 and 2 score that has better prognosis. So, and what is actually very nice is that IDH1 and 2 are moderately abundant proteins that can likely be quantified directly by targeted mass spectrometry, MRM mass spectrometry assay without enrichment. So it will be, it can be used in clinical practice. So another thing we did is the drug analysis for high-grade glioma. Here we don't have normal tissue, but what we did is to capture the aggressiveness of high-grade glioma compared to low-grade glioma. So basically comparing high-grade glioma to low-grade glioma, we build up a signature. We found genes, protein, and phosphoprotein differentially expressed between the two diagnoses. And then considering the set of genes, protein, phosphosites upregulated in high-grade glioma, we leveraged the link CMAP L1000 transcriptomic database and P100 proteomic perturbation drug analysis database in order to find the drugs able to reverse that particular signal. So basically what we found is that CDK inhibitor drugs were at the most at the top of the list. So they were the ones that were able to basically reverse this aggressiveness signature. And basically the signal was also confirmed by the protein data. So what this heat map show is the basically if the CDK1 was upregulated in high-grade glioma compared to low-grade glioma. As you can see, RNA and also proteome data are basically capturing this upregulation. Also, what we did is an investigation based on phosphorylation data. So this box plot captured the kinase activity score. So for each kinase, we look at the phosphosites targeted by that particular kinase, and based on their phosphoabundance, we construct a signature. And basically that signature, as you can see, so the phosphosites targeted by CDK1 were upregulated in high-grade glioma compared to the low-grade glioma. The same for CDK2 and also MAPK1. In fact, the MEK inhibitor was another drug that was scoring at the top of the list, able to reverse the aggressiveness of this tumor. So this is the first large-scale proteogenomic study of pediatric brain tumor. So the few highlights are the new treatment strategy for craniopharyngioma. We found a subset of craniopharyngioma BRAF wild type with a similar proteomic and phosphoproteomic profile to the low-grade glioma carrying the BRAF V600E mutation, and this suggests the potential use of MEK and MAPK inhibitors. And we found a new prognostic marker for high-grade glioma. We also developed a web platform to facilitate the navigation of our protogenomic data. I have a slide for an overview of the platform. So basically, every user can input any gene, and all the data that we have from copy number variation, mutation, proteome, phosphoproteome data for that particular gene is going to be displayed. And on the top of the heat map, you are going to have the diagnosis, so different histologies, tumor location, grade, and all the clinical data. So you can basically sort based on each variable, and you can also zoom in and consider just one histologies at the time. So this is going to facilitate the exploration of the data, and I hope that people can explore our data using this user-friendly platform. So as future steps, so we published this first protogenomic characterization of pediatric brain tumor in Cell, and then we also did a glioblastoma, adult glioblastoma protogenomic characterization that was recently published in Cancer Cell. And now we are working, the CPTech and CBTN are working together in order to generate data for AYA data, so adolescent, young adult data, to link the pediatric and the glioblastoma study. And also, as I mentioned previously, Adam, Brian, and Amanda Pavlovich, they submitted a DOD for the craniopharyngioma clinical trial in order to test the MEK inhibitor therapy for craniopharyngioma not carrying the BRAF V600E mutation. So I needed to thank so many people that contributed to this study. I want to thank Henry Rodriguez from CPTech, NCI, Adam Resnick, of course, and Brian Rudd and Pei Wang, and all the people that contributed to the study. Thank you so much.
Thanks so much, Francesca. And while Valerie switches the share screen, let me just take an opportunity to really thank you and and the team at Mount Sinai for really, I think, carrying the spirit of Kids First, which is integrating across modalities and domains. It's been one of the most fruitful collaborations of my own career, and I know especially, on behalf of the pediatric community, your engagement, the pediatric landscape, coming from the adult side has been phenomenal. Let me just take 5 minutes to, and folks, if you have questions for Francesca, please feel free to put it in the Q&A, because I know we're going to potentially be short on time. Let me share my screen and start here. One of the things that made the proteogenomic analysis with CPTech and the team so amazing, because it really began, pushing on the limits of thinking about how to support discovery across not only different domains, but also different resources. And I want to just highlight, where we're heading in this context, because all of the deployments that Vincent highlighted and some of the work that you've seen really begin to move us, as I've described before, across this essentially funnel for discovery. We really are getting to this knowledge to impact context in the queryability context, but also, as you can see from the amazing work that was presented earlier today and just now with Francesca. But the challenge that you begin to encounter as someone who's trying to build platforms on behalf of discovery is this constant need to think about vertical integration across modalities within your own platform or within the pediatric context. But as Francesca highlighted, also think about how do you connect and leverage other resources across other environments or other research contexts in this context of horizontal integration. And this is a perennial challenge for the pediatric community as a whole, because the landscape of discovery in pediatrics spans, all institutes and centers at the NIH, and pediatric cancer and structural birth defect data really essentially occurs across many contexts of development. We've highlighted that in this platform development context, our attempt to also address the very diverse community of stakeholders that are required to put all this information together, ranging from the coders and developers all the way on the right of the spectrum to people like myself, who are much more reliant on clicking our way through hypothesis testing on the portal context. And what we're seeing now is the capacity to look at that dimension across users, not only across cancer and birth defect, but then also across modalities of clinical data, whole genome sequencing, RNA-seq, proteomics, and imaging. And this really presses on the community engagement and opportunity context. There are many dimensions to this challenge and opportunity, and one of them that we're especially excited about from the very beginning of Kids First has been this multi-platform context that spans across some of the ongoing work that's occurring under the NCPI initiative led by the NIH across Kids First, the Anvil, Biodata Catalyst, and NCI, and the Common Fund data ecosystem. And I wanted to just share a couple of seconds to show how the Seven Bridges team really supported this new context for cross-platform interoperability in ways that addressed really how the science was done. Specifically, the Kids First data resource was initially the host for the genomic data for this proteogenomic characterization, and the Proteomic Data Commons is the host for the proteomic data. But as users, you want to be able to traverse and intersect these data in really meaningful ways. And as platform developers, it's one of the challenges that we have to support. And I'll just show you a few seconds around how that process is done, and then we can finish. So as I mentioned, all the proteogenomic data, associated genomic data is in the context of the Kids First data resource, which we've reviewed in the past, where users can explore and find, for example, all brain tumor data. Here you can see that I already have my own saved query across the proteogenomic cohorts that includes 192 of those subjects and the associated genomic and clinical data. All of this data is also ingested into the associated PTC bioportal environment, where users not only can review the at least called mutational context from the Kids First data resource, but also can view the proteomic data in this context as associated. And here I just queried for BRAF within the context of the mutational spectrum across the craniopharyngiomas, for example, and low-grade gliomas is the cohort I chose here. We also are thrilled with opportunities for other developers, like Francesca mentioned, to develop additional portals or views around these data, especially when open access. And this is really an amazing and intuitive platform that Francesca highlighted around the Cell paper, and for users to really query the actual data in ways that are very similar to the figures in the paper itself, where you have the associated clustering that the paper defined. But if a user wants to bring the source data from proteomic data and the source data from Kids First together for analysis in a computational environment, how does one do that? And so I just wanted to briefly highlight a new integration and deployment supported by Seven Bridges. So if you go to the Proteomic Data Commons, you can find, for example, the pediatric AYA brain tumor cohort within the PDC. And you can essentially review the entirety of the cohort in this context across a biospecimen, clinical, file, and gene context. And if you go to the file context, you can see essentially every file that's produced as part of the project, including the tables that annotate the samples and phenotypes. As a user, if you want to actually engage in your own analysis of a particular proteomic file, all you have to do is actually just download essentially a manifest of any file of interest from the PDC by clicking on this button, and this will generate a CSV file. That CSV file can then be directly uploaded into the Kavatica environment. So we've shown in the past how Kavatica is integrated into the Kids First data resources such that users can push their genomic data into a project. But now we're showing how a user can leverage these manifests in an easy way to upload files. So I'll just show you briefly that you can essentially click on this import manifest context here. You'll have a choice to do this from the Proteomic Data Commons in this context here. You can select the file of interest that you essentially just downloaded. and then import those files. And in doing so, those files will show up within your Chematica environment alongside any genomic files that you chose to bring in from the Kids First environment, should you have done that. Also, over the past week or so, the Summer's team additionally deployed tools that support proteomic analysis, including common tools like the MaxQuant environment, for analysis of both the raw files and associated input FASTA files for your own integration of either proteomic data from CPTAC, which is also all available on Chematica. Indeed, that's one of the highlights and recurrent themes for us, is that within places like Chematica, you can bring together target data, the proteomic, genomic, and other files from different platforms, and integrate them with both adult and pediatric context data in ways that can be reproduced across the public app space in continuous integration of publicly available workflow language encoded tools. So I'll pause here and turn it over back to Valerie, but really kudos to Francesca, Pei, and all of our collaborators, including those at Fred Hutch and many others, for supporting really this cross-platform and cross-age integration of data.
Thank you so much, Adam. And we are running short on time, so unfortunately I am going to take back the control, but I did upgrade a lot of my NIH colleagues to panelists, so while I'm talking, they can all help me answer any questions that come up, and please feel free to continue asking questions in the chat. With the time that I have left, let me give you some higher level updates about what's been going on with Kids First. It's just been an insanely busy year, so we decided to organize this according to some of the key questions we received from you all in advance of the webinar. So the first question was, what are priorities for Kids First moving forward? And as you've hopefully seen, researchers are really using the Kids First data resource. They're accessing the data in the portal. More than 2,100 researchers from across the country and the world have already registered with the portal, where you can view those data sets, interact with those data, and soon you'll be able to search variants there as well. And across the 19 data sets that are released, there are over 200 approved dbGaP data access requests, which means that those researchers are accessing the individual level controlled access genomic data and actually doing those in-depth genomic analyses like what we saw from Dr. Engel's group today. And this list of 230 actually does not include the X01 investigators who contributed the cohort or their direct collaborators. It is the end users, the other researchers who are accessing these data. So that shows us that the data resource is really strong. It's been of high value to the pediatric research community. And for the remaining years of the program, the goal is to build on that infrastructure, the data sets, and the expertise that all of you are bringing to the table in this Kids First community. And the goal is to leverage all that to enhance the program's impact in pursuit of improved preventative measures, diagnostics, and therapeutics. We intend to do this through 3 broad initiatives, including additional data generation of childhood cancer and structural birth defects data sets. We do plan to continue some whole genome sequencing, but we're also looking at adding some of the epigenomic assays and proteomic assays, both of which were actually highlighted today. We plan to continue to improve and develop the data resource, not doing it alone, engaging many researchers and helping us do that, and to start looking for ways to sustain how the resource and its impact will continue beyond our 10 Common Fund years. We also plan to engage experts in a variety of activities to increase the value of the Kids First data sets, whether that means integrating new data types into our existing projects like imaging or richer clinical and phenotypic data, or again, integrating new tools into the environment. How are researchers using Kids First data? So there are multiple answers to this question, multiple places you can go to learn more. So every genomic data set is associated with a dbGaP study page. Remember, researchers are required to get dbGaP approval before accessing individual level genomic data. And when they do that, they have to tell the NIH what they plan to do with it. And that information is actually made publicly available on each dbGaP page associated with the study. And I know I'm running out of time, but I'm going to show you an example so that you know you can get this information. So if you're on a dbGaP study page, you can click on this tab called Authorize Requests, and that will just jump you down. And it shows you how many researchers, how many principal investigators have applied to access this data set. It shows you who they are, and you can see their research use statements, both a technical research use statement and a non-technical summary of their work. So that is definitely a resource that's available for you to look at if you're really interested in knowing what researchers are actually doing. We also wanted to update you that we have noticed a proliferation of NIH grants that are being used to analyze Kids First data sets. Now, many of you are familiar that we have a Kids First R03 PAR that was specifically geared towards fostering analyses of Kids First data sets. And 19 awards have been made to date, which is pretty exciting. But researchers are also using other FOAs, other grant mechanisms to, leveraging the resource of the Kids First data that's available to advance a variety of projects, including even clinical trials and some training awards. So really exciting to see that very recently the research is a variety of research projects are being done on the data that's been shared. We've highlighted in the past that the data has also inspired some collaborations with the Knockout Mouse Phenotyping Program and the investigation of co-occurring conditions across the lifespan to understand Down syndrome project. We also wanted to make you aware that all of this information about Kids First, or not Kids First, and all NIH grants are publicly available through a resource called the NIH Reporter. So if you're really interested in seeing investigators who are funded, what their project abstracts are, this information is fully available to you. And if you're specifically interested in Kids First projects, we recommend searching with the terms Gabriella Miller, Kids First, and or looking for a specific investigator or the project you're interested in. So that's definitely a good resource for you to see exactly what the projects are. We also have a publications page where we are on a regular basis learning about the papers that are being published based on findings in Kids First data sets. And recently this got up to 22 publications with a relative citation ratio of 1.40. And I could not fit all of the publications on this slide, but we certainly encourage you to visit that link to check out all of the papers to date. And of course, we use these webinars as a venue to tell you about some highlighted projects and work that is going on. All of this is available on our new meeting page. All of the past webinars will be there. How can I connect with other Kids First researchers? The DRC's website and portal actually has some resources for this. We have our classic X01 page that we host on the Kids First Common Fund page, but you can also see profiles on the Kids First DRC's page, where you can see the different investigators, including members of the DRC, as well as the sequencing centers. You can just read about the investigators who contributed these cohorts. When you're in the portal, you can also reveal, when you register for the portal, you probably notice you do have to make a little light profile, but usually that's hidden from other researchers. If you set that status to public, that will actually expose your profile to other people in the portal, and you can search, that means that people can search for you or find you in the portal itself. And unfortunately, I don't have time to demo that. But I do want to tell you just before we end about FOAs that are available. We have a new RFA, Expert-Driven Small Projects to Strengthen Gabriella Miller Kids First Discovery. This is an R03 RFA, so slightly different from our existing R03 PAR. We recently held a pre-application webinar, and I encourage you to watch that if you're interested. The goal of this FOA is to engage experts in a variety of activities to enhance Kids First data and resources. So a little bit different than an analysis focus, which is what the other R03 is for. We have listed FOAs on our website. We have an FAQ page, but we've also compiled a list here. We also compiled a list of FOAs that might be useful to you if you're looking to do that next step, that variant validation step. How does Kids First collaborate with other efforts? We've talked about this a lot in the past. In the last webinar, we told you about our cross-platform collaboration, and Adam mentioned this as well, to have platform interoperability and enable researchers to co-analyze multiple data sets. But I do want to take a moment to highlight the recent collaboration with the Monarch Initiative on the Innovation Across the Phenotypic Translational Divide webinar series. Parts 1 and 2 were held earlier last year and brought together a lot of researchers to talk about challenges in phenotyping data sets across Kids First and how to enable analyses, how to make those data sets stronger. We recently hosted a part 3, which was focused on cross-species analyses, so talking about how model organism data sets and databases could be linked to the Kids First data resource and Kids First data to inspire new types of analyses and understand these conditions better. We have a new FAQ to address a question about what standards are available. This includes data standards, technical standards, standards about different types of data. There are certain data standards that the Data Resource Center has adopted to make data more easily cross-analyzable or more findable across the data resource. But there are also other standards that NIH recommends or different specific research communities recommend, and we try to compile that all for you on the FAQ page.